All right, let's dive in. Today, we're going to decode the science and the business behind DNA sequencing. We'll start big, with the massive multi-billion-dollar market it's created, and then we're going to zoom right down to the lab bench to see exactly how scientists use this incredible technology step by step.
So just how big is this market we're talking about? Well, let's start with a number. In 2025, the global DNA sequencing market is valued at a pretty staggering $14.7 billion.
But hold on, that's just where we're starting. The growth here is, well, it's explosive. By 2034, that market is projected to more than triple, rocketing past $51 billion.
What does that mean in terms of speed? It means this market is expanding at a compound annual growth rate of nearly 15%. That is a blistering pace for an industry this huge.
So what's fueling this genetic gold rush? Well, it's really a perfect storm, right? You've got these huge leaps in technology crashing right into this massive, growing demand for things like precision medicine, where treatments are literally tailored to your own DNA.
At its very core, DNA sequencing is all about information. Think about it. This is the technology that lets us read the fundamental code, the actual blueprint of any living thing. The sheer amount of data this generates about our genes is changing, well, everything.
So what's the engine driving this whole revolution? It's got a name: Next Generation Sequencing, or NGS for short. This is the core tech that has made sequencing faster, cheaper, and way more accessible than anyone ever thought possible.
And this is where things get really wild. Look at the sheer scale of this leap. The classic method, Sanger sequencing, was chugging along, doing maybe a few hundred DNA pieces at once. NGS, it's on a whole other level. We're talking millions of fragments at the same time. This is what just blew the doors wide open for genetic research. So because it's so fast, so massive, and, crucially, getting cheaper all the time, NGS has gone from this highly specialized tool to something you find in labs and clinics everywhere. It's totally transformed pretty much every corner of genetics.
Okay, so that's the big picture. Now let's get our hands dirty. We're going to zoom all the way down to the lab bench. Think of this next part as our own virtual lab session. We're going to walk through how a scientist tackles a really critical problem, making absolutely certain that a piece of DNA they've built is exactly what it's supposed to be.
And trust me, the stakes here are incredibly high. Just look at this quote from a 2020 paper. It says, researchers are often tempted to skip this verification step because, let's face it, it can be a pain. But when they do that, it can cause huge problems with reproducibility, meaning the whole experiment might be a wash, undermining science itself.
So here's the problem scientists are up against. In modern biology, they're building these huge libraries of custom DNA circles called plasmids. We're talking hundreds of them. Trying to check every single one with the old Sanger method? Forget about it. It would take forever and cost a fortune. They desperately needed a better way.
And the solution, laid out in that same paper, is, well, it's just brilliant. It's this three-step open-source computational pipeline that uses the power of NGS to do the job fast and cheap. So let's walk through it, like we're running the experiment ourselves.
The first step is actually pretty counterintuitive. You see, NGS just floods you with data, and you'd think more is better, right? Well, the researchers found that too much data can actually mess up the assembly. So the pipeline is super strict. It just throws out most of the data, keeping only the absolute best of the best, like the top 3 to 11% of the sequencing reads.
Okay, so this next step is the real magic. It uses something called de novo assembly. Basically, instead of checking the DNA against a map you already have, it builds the map from scratch. It's like putting together a jigsaw puzzle without ever looking at the picture on the box. And why is this so powerful? Because it means you can find the real sequence, even if it has mistakes or mutations you never, ever expected. So to do this puzzle solving, the pipeline uses an open source tool called Unicycler. It takes those top quality reads and digitally stitches them all back together to rebuild the complete circular plasmid. And get this, it's unbelievably fast. We're talking less than 8 seconds, on average, to reconstruct an entire plasmid. All right, last step. This is our final quality check. The pipeline takes all that original data and basically lays it on top of the new sequence it just built. This does two things. First, it makes sure everything lines up perfectly. And second, and this is super clever, it can actually spot if your original sample was contaminated with any other random DNA. Okay, lab coat's off, lab session's over, let's zoom back out. Because that super detailed process we just walked through,
That's not just some random lab exercise. That's the bedrock. It's the kind of fundamental work that makes all the advanced stuff possible. So how does this connect back to that $50 billion market and, the future of our health?
And that brings us right to the biggest driver of this entire market. Look at this. The biggest piece of the pie, by far, belongs to oncology. That's cancer research. Doctors and scientists are using sequencing to read the DNA of tumors, find the exact genetic glitches that are causing the cancer, and then pick the perfect targeted drug to fight it.
And it all comes full circle, right? It ties back to everything we talked about at the start. You've got the huge demand for this kind of precision medicine, the sad fact that cancer is more prevalent, tons of money pouring into R&D, and of course, the cost of sequencing just keeps dropping. It's all feeding into each other.
And this isn't just happening in research labs. It's being woven right into our healthcare systems. You have entire countries launching massive projects to sequence their populations, creating these incredible databases to help us understand diseases. And you also see it in things like newborn screening, where we can spot the risk for genetic diseases the moment a child is born. That means we can intervene before they even get sick.
And all of this leads to one final, really big question. The cost to sequence a single human genome is just plummeting. So as this technology gets cheaper and more powerful and just everywhere, you have to wonder, how is our ability to read our own DNA going to change the very definition of medicine itself?